And we will start with the role of calcium in muscle contraction. So when you contract your muscle, what are the requirements for the muscle contraction? So we need ATP, calcium, ions, and nervous stimulation for the muscle contraction. Okay. So calcium in the muscle is stored in the sarcoplasmic reticulum. Sarcoplasmic reticulum is modified smooth endoplasmic reticulum in this muscle fiber. And the role of calcium is when calcium binds with troponin, then it moves the tropomyosin from the binding site where myosin head can bind. So you can see in the relaxed muscle, you see this is relaxed muscle and this is contracting muscle. In the relaxed muscles, you have actin, myosin, myosin head, then you have troponin, and you have the long thread of tropomyosin. And you see here in relaxed muscle, tropomyosin is covering the binding sites. Once calcium, after the action potential, enters the muscle cells, and calcium released from the sarcoplasmic reticulum in the sarcoplasm and binds with the troponin, then what it does, you see, the tropomyosin is moved and now myosin head can bind. Okay, so in contracting muscle, myosin heads binds to actin. In relaxing muscle, it cannot because it is covered by tropomyosin. So when calcium level rise in the sarcoplasm or cytoplasm of muscle cell, which comes from the sarcoplasmic reticulum, calcium binds to troponin, causing conformational change in the position of tropomyosin, which moves tropomyosin and exposes binding sites. That allows cross-bridge and contraction to occur. And cross-bridge cycle stops when calcium level decrease. So once muscle contraction happens, then what happens? This calcium again released back. This detached from this troponin and comes into the cytoplasm. And then another ATP is needed to put this calcium ions back into the sarcoplasmic reticulum. Okay. So to show you where calcium is in the muscle, you can see here. This is a muscle cell, a muscle fiber. You have the organelle myofibrils inside the muscle cells. And then the plasma membrane or sarcolemma get inside the pink tube. It goes around the myofibril. So it wraps around pretty much all the myofibrils. And linked to these transverse tubules, you have the green tubules that is called terminal cisternae. and they are enlarged part of sarcoplasmic reticulum. These tubules stores calcium. So let's see, when your motor neuron release neurotransmitter on this neuro junction on the plasma membrane of this muscle fiber, the action potential travels all the way through these transverse tubules around the myofibril, and that opens the calcium channel here and release calcium from the cisternae of the sarcoplasmic reticulum and spread around and inside the myofibril, which ultimately binds with the troponin. Okay. Once, due to the crust bridge sliding filament theory, once action potential is taken back, depolarization, then again the calcium detached from the troponin and pumped back into the sarcoplasmic reticulum using the ATP, that is active process. Okay, so calcium level decrease because it is continually pumped back into sarcoplasmic reticulum, which is a sarcoplasmic reticulum is a calcium reservoir in muscle, and most calcium in sarcoplasmic reticulum is in terminal cistern, in this dilated part of the sarcoplasmic reticulum. They run along the side, and these three structure you see here, two tubule, sorry, cisternae and one tubule. Together they are called triad. Triad of myofibril. Okay.
So that includes the SR, the T tubules, and the terminal cisternae, all three of those?
Yes. So two terminal cisternae, you see, and the T tubules. That's how they are three. This cisternae, and T tubule. Okay. So let's see here in detail the same thing. Excitation, contraction, coupling. So excitation is electrical activity and contraction is physical activity in the muscle. Excitation you don't see, it is impulse. Contraction you see, when muscle contracts, you see the physical movement. So when motor neuron brings the action potential at the nerve terminal, action terminal, they release neurotransmitter through the vesicle, and these neurotransmitter then binds with the, you see, number one, nicotinic acetylcholine receptor on the skeletal muscle plasma membrane. Number one. Number two, skeletal muscle voltage gated sodium channel open. So you have the green here, the blue one, two. These are also another channel in the plasma membrane of muscle fiber. When these voltage gated sodium channel open in response to binding acetylcholine with the acetylcholine receptor, where is sodium more? Sodium is more outside the cell, so sodium get inside the cell. And that depolarize these muscle cells. Once it depolarize, the depolarization of the muscle cell also travel all the way inside the cell and around the myofibril through the T tubules. You see, this is the T tubule. Transverse tubule voltage gated calcium channel. So you see some of the calcium, then there is calcium channel. Some of the calcium from outside also moves inside the cell, and that helps sarcoplasmic reticulum calcium release channel to release calcium from the cisternae into the cytoplasm. You see, calcium is coming outside. And these calcium then binds with one of the protein in the sarcomere. Which protein these calcium ions bind with?
Tropomyosin.
Does it bind with tropomyosin?
Troponin.
Troponin. It binds with the troponin and then it moves the tropomyosin. Okay. So that's how this is the excitation up to here. And then when these actin and myosin when slide with each other and sarcomere decrease in length, that is contraction. And these couples with each other. So contraction couples with excitation. That's why we call them excitation-contraction coupling. So muscle contraction, what are the essential element factor of muscle contraction?
ATP.
Of muscle contraction. Let's check the memory from the past. So you said ATP, one.
Calcium ions.
Calcium ions,
Calcium ion,
nervous stimuli,
Nervous system stimulation.
okay, nervous stimulation, so action potential APs, acetylcholine. What else? Do you
Sodium.
need- yes, sodium ions.
Potassium.
Potassium, good. Okay, so let's summarize all our electrical impulse too then. So where you need calcium?
To take it to the terminal, right? To bind with the,
Okay, one calcium is here.
to
Do
release.
you need anywhere else?
So that it can bond to the troponin and allow the actin to bind to the troponin that allows it to go on the head.
That's the one role of calcium. Is there any other role of calcium?
It allows the acetylcholine vesicles to bind.
Very good. To release the acetylcholine from these nerve terminals, first calcium needs to get inside the cell. You remember that? In nervous system, we did release of neurotransmitter. Okay, that two location, you need calcium. Okay. Now, where you need ATP.
For the energy.
Yeah. Again, to take back that acetylcholine, once acetylcholine is released back here, to pump it back, acetylcholine in the nerve terminal, you need ATP. To bind myosin head with the actin, you need ATP. Calcium is released here. To take this calcium back, once the muscle contract and once the muscle relax, you need to pump back this calcium back into the sarcoplasmic reticulum. You need ATP. The sodium enters the cell for the action potential. Again, to bring this muscle fiber to the resting membrane potential, again, this sodium is back extracellular, you need ATP. So you know how much benefit is if you exercise. You are using a lot of ADP. That's the ATP needed for the muscle contraction and other actions. Action potential, you know, you need acetylcholine. Where you need sodium, you see here, sodium ions. Where you need potassium, to generate action potential anywhere you need in the nerve fiber. Sodium is getting in, potassium is getting out, so you need sodium, potassium. This is in detail to understand the real physiology, okay? So keep in mind these things all the time. That's why electrolyte, these all are electrolytes. Sodium, potassium, calcium, and we need all the time in our body. So let's see if you have sodium, potassium, calcium deficiency. What will happen? You will have physical fatigue or psychological fatigue. Sometimes what happens, you don't want to walk, you're lying down, you know you have to go to exercise, but you say, no, I don't want to. Do you know why that happens? Due to these the electrolytes deficiency. That's called psychological fatigue. And sometimes you exercise and then a lot of the lactic acid build up in your muscle, which is called lactic acidosis, and muscle is fatigue. That is real fatigue. You need rest in that case. Okay. So that is the excitation contraction coupling. This is the excitation contraction coupling steps we talked about. See, as you see, somatic motor neuron. Somatic motor neuron is motor neuron which is innervating or supplying the skeletal muscle cells, muscle fibers. So these somatic motor neuron from the motor unit release acetylcholine. That acetylcholine then binds on the sarcolemma, the plasma membrane of the muscle fiber, to the nicotinic receptor, which opens ligand or chemically gated channels. That means those chemically gated channels are sodium channels. One sodium channel opens, sodium diffuse in the muscle cell, producing depolarization stimulus of the muscle cell plasma membrane. Action potential produce. That action potential then get inside the muscle cell and around the myofibril through the transverse tubules. So action potential conducted along transtubules to reach the myofibrils and to reach ultimately the actin and myosin. Action potential open voltage gated calcium channel in the plasma membrane of the muscle cell, and that then activates sarcoplasmic reticulum to release calcium from sarcoplasmic reticulum, and then calcium diffuse out in sarcoplasm, which binds to troponin and stimulates contraction. Okay, so this is the process by which action potential cause contraction. Okay, muscle relaxation. When any work we do, we think when we do muscle contraction, we use ATP, but muscle relaxation also needs ATP. If I ask ATP needed for muscle relaxation, why? Then what is the answer? How muscle relax? Muscle relax because
An ATP.
the bind the myosin head detach from actin using the ATP. Yes, once another ATP bind, then they detach for relaxation. Yes, that's why you need ATP. Where do you need another ATP for muscle relaxation? Action potential back, sodium, you have to put the sodium back, you need ATP. You have to put the calcium back into the cisternae, you need ATP. So every step in muscle relaxation, you need ATP. And that's why even muscle relaxation is energy requirement. Okay. So calcium ions from sarcoplasmic reticulum diffuse to troponin to initiate cross-bridge cycle and contraction. When action potential ceases, muscle relaxes because calcium channels close and calcium is pumped back into sarcoplasmic reticulum by calcium ATPase pumps. Therefore, ATP is needed for muscle relaxation as well as contraction. That's why muscle contraction has a double benefit. Okay, now the contraction of muscles, which is the mechanics of muscle contraction. Let's see here now. We're going to talk about twitch, summation, and tetanus. So let's go back. How muscle is innervated. So a single motor neuron comes, branches, and then each smaller branches ultimately make a neuromuscular junction with the muscle cell. So once a motor neuron release neurotransmitter and contract one single muscle fiber, that is called twitch. Have you experienced like your twitching of your eye, single twitching? That is like kind of a twitch. So a twitch is when a single rapid contraction and relaxation of muscle fiber happens. Okay. So you see here, this is the myogram, and this is the electrical muscle contraction here. So when you give a stimulus, once the stimulus is adequate, you have a single muscle contraction. And then what happens? There is relaxation. But if you give, so what happened? You have muscle contraction, you have calcium inside your cytoplasm. And then if we don't give immediately, what will happen? There will be big relaxation phase, this one. Okay, so we are not giving, so here is relaxation phase. And if you give immediate another stimulus, then what happens? The second stimulus is piggybacking on the first one. And it is stronger than the first one because you did not allow calcium to go back into the sarcoplasmic reticulum. And that is called summation. Okay, so if second stimulus occurs before muscle relaxes, means even before calcium is back into the sarcoplasmic reticulum, from first, the second twitch will be greater, and that is summation or addition of the twitch. Okay. Contraction of varying strength, graded contraction are obtained by stimulation of varying number of fibers. So if you see, let's see, if I give second one, third one, fourth one, fifth one, continuous stimulation, then what happens? It's going to be stronger and stronger and stronger. And then you're going to see like a straight line. That is called tetanus. So you see here. Let's see here, there is like impulse when you do like muscle myogram or muscle contraction exercise. When you give first twitch here, what happens? You have first twitch, the relaxation a little bit, second twitch, third, 5 shocks per second, and then you increase. Now you give 10 stimulus per second. You see, there is no relaxation. And this is called incomplete tetanus. If there was single one, then we call them twitch. If there is single and then next bigger one, that is the summation. And now, if you increasing the shocks like 60 shocks per second, what happens? Like sustained muscle contraction. This is called complete tetanus. But does it remain forever? No. Why not?
You get tired, or you get tired,
Muscle fatigue.
or
Why you have muscle fatigue?
Lactic.
I think the calcium, well, the troponin leaves the site, it's taken back by ATP. Then you have to.
So what happens? Again, you are depleting the ATP.
Right.
You have not enough calcium.
Mm-hmm.
And or you have a lot of lactic acid buildup, and that gives the fatigue. Okay, so this is called sustained contraction. Tetanus, don't get confused with the disease tetanus. This is sustained muscle contraction. Okay.
Professor, I have a question. So is it only shocks that can cause tetanus, or like what's a real life event that can cause tetanus?
Or this is just a SOX means you are giving, when you do this lab, you do the SOX. Okay, you give the electrical shock. But in real life, what happens? You contract, like when I'm lifting, let's say, heavier thing. So when I lift, like I'm lifting this cell phone, and when I try to lift this table, what happens? First, I cannot lift, and then I increase, recruit, and then I lift all. And when I'm lifting that whole table, that is tetanus, because all the muscles are in contracted form. Sustained contraction, continuous contraction, that is tetanus. Are you following me?
Yes, thank you.
This is the lab portion, that's why we call shock. In real life, our brain knows it how to do it. They send the electrical impulse from our brain through the motor neuron and keep the tetanus continuous.
So
Okay?
basically, it's like the keeping the contraction on like a hold.
Yep. Yep.
So a tetanus isn't necessarily a bad thing, right?
Yeah. So tetanus is not bad thing. That's why I said earlier, don't get confused tetanus with the disease tetanus. Disease tetanus is bad because your muscle is contracted because you have sustained calcium, because and there is a lot of acetylcholine coming because you enzyme is degraded in disease, and that's why there is a lot of acetylcholine, and your muscle is like abnormally contracted even when you don't need to hold anything. That's the disease. This is when you need it and you are working it. Am I clear?
Yes. So professor, so someone who's deficient in calcium, they wouldn't be able to lift up the table at all, right?
Yep.
Or, okay.
Yep. Okay. So if muscle is repeatedly stimulated with maximum voltage to produce individual twists, successive twitches get larger, and this is called trippy or a staircase effect. And this is caused by accumulation of intracellular calcium. Like you are building, you are not letting the calcium go back. So you have tetanus, and if you give even extra stronger, then you have a stronger and a stronger, like the staircase effect, and that is called trippy. Okay, now energy requirement for skeletal muscles. So before we go and detail, what are the source of energy for the exercise or skeletal muscle contraction?
ATP.
ATP,
ATP. Where you get that ATP from?
mitochondria,
Mitochondria.
Where mitochondria get that from?
food,
What kind of food?
sugars,
Carbohydrates.
Carbohydrates.
Protein.
Protein, fat, anything else?
Some alcohol is technically one.
Alcohol, not good source. Anything else? Creatinine. Have you heard that the athletes pump creatinine?
Yeah.
Yeah. So creatinine.
Yes.
Because creatinine phosphate has phosphate, and that creatinine first 1 minute, around 1 minute to 1.5 minutes, to build your respiratory system, cardiovascular system, to provide energy for the exercise. Immediate energy come from that one. So if somebody is like 100-meter sprint, in that case, creatinine is very important. More the creatinine, faster you can work. Okay. So let's talk about a little bit about the energy requirement of a skeletal muscle. Okay. Metabolism of skeletal muscle. Skeletal muscle respire anaerobically for first 45 to 90 seconds of moderate to heavy exercise. That means during that time, your muscle can get ATP either from the creatine phosphate, because creatine phosphate provide phosphate to ADP and then forms ATP, okay? Or your muscle cell will do glycolysis. Glycolysis means they take glucose or glycogen, which is the storage of chain of carbohydrate in your muscle, and break down them into pyruvic acid and then make some ATP.
Is that the one where it skips the two other cycles, where it skips the citric acid and the
Yes.
That's what that is?
Yes, because citric acid cycle takes long time. You take pyruvic acid to the mitochondria and then you make ATP, but you will make more ATP. But glycolysis is you make ATP fast because you need immediately. Your Krebs cycle or oxidative phosphorylation is not active at. Okay, so that's it there. So in that time, what happens? Cardiopulmonary system requires this time to release O2 supply to exercising muscle. If exercise is moderate and it continues, then aerobic respiration contributes majority of muscle requirement after first 2 minutes. So if you want to burn the fat in your body, you need to do moderate exercise. Moderate exercise burn most of the fatty acid through the oxidative phosphorylation or Krebs cycle. Are you following me? If you do heavy exercise, running fast, you're going to take most of the energy from glucose by beta oxidation. Okay. So during light exercise, most energy is derived from aerobic respiration of fatty acid. During moderate exercise, energy derived from equally from fatty acid and glucose. But during heavy exercise, glucose supplies 2/3 of energy. Because during heavy exercise, liver has a lot of glycogen. They increase glycogenolysis, breaks down glycogen into glucose. And then GLUT4 transporter. GLUT4 are the channel which put into plasma membrane of a skeletal muscle and takes glucose from blood into your muscle. And then muscle takes it to the cytoplasm or into the mitochondria where they make ATP by a glycolysis or Krebs cycle. You know, glucose transporter 4 is the receptor which carry not impulse, sugar from your blood into your muscle cell. And to function this GLUT4, you need insulin. That's why muscle cell, fat cell, and liver cell need insulin to take glucose inside the cell. So if somebody is type 2 diabetes, they will not take glucose enough for their muscle. That's why they feel tired all the time. They cannot use that glucose for the source of energy, and glucose remain outside. Okay. So you can see in this diagram, this is the percentage of energy expenditure during 30 minutes of exercise. Muscle glycogen, our muscle stores glycogen in the form of big chain of carbohydrate, muscle triglycerides, plasma free fatty acid, and plasma glucose. So mild exercise, most of the energy is coming from
Fatty acids.
fatty acid. So let's say if you're jogging, sorry, you are walking, you walk like 1 hour, you burn a lot of fat. Moderate exercise like mild running or fast brisk walking, still you do a lot of the free fatty acid, but most is muscle glycogen. If you, let's say somebody is sedentary and don't exercise, this muscle glycogen, you know, in the muscle, what they do? They start turning into fat.
Fat droplet.
Yep. If you have a heavy exercise, you are breaking down muscle glycogen, and indirectly you are burning out fat too. Okay. Now, percentage of energy expenditure after half an hour. Okay. So you see here, plasma free fatty acid, if you walk longer, then fatty acid is going a lot. So that's why every weekend we should walk at least 2 hours.
So quick question, Dr. Shah.
Uh-huh.
So as far as like if someone's just trying to like lose weight purely to lose fat, would you say like it would be better for them to just do like an hour-long walk or like an hour long of heavy exercise, like going to the gym and doing weights and like doing a bunch of running? Like, would it be more beneficial if they did longer periods of mild exercise than shorter periods of heavy exercise?
Longer period of mild exercise.
Okay. Gotcha.
Yeah, and the heavier exercise always works, because indirectly what happens, we're gonna burn muscle glycogen, so that will have indirect effect. But if somebody has high cholesterol, I will suggest do this one.
The longer period of mild exercise. Gotcha.
Yes, yeah. And something came to my mind. Okay. If it comes again, I will tell you.
I'm sorry to get you off track. I was just curious.
Oh,
and
yeah,
I
something was
apologize.
going. No problem, no problem. So this is the exercise protocol when you're doing. Like, longer exercise burns a lot of fat, but heavier exercise provide more oxygen, and then they help in other organs to burn more energy. So that is another part. It's not only muscle burn our energy, there is other parts of the body too.
Professor, so since you're talking about this, heavy exercise, so doesn't that increase the muscle mass? Like you get a bigger.
Yes, very good question. Yep. So when you do, let's talk about the muscle cells. Muscle cells are long, thin filament. Yes. And inside the muscle cell, you have nucleus, you have sarcoplasm, you have sarcoplasmic reticulum, and the most important, you have myofibril. So what is the difference between a person who exercises and who does not exercise? When you exercise, you don't increase the number of muscle cells. What you increase? You increase the amount of protein, which is actin, myosin, troponin, and tropomyosin. So you increase the bundle of myofibrils inside your cell. And that's why your cell size becomes bigger. And that's how your muscle is trophy and a stronger muscle you develop. In exercise, you increase mitochondria number. You increase number of mitochondria because mitochondria has their own DNA and they can divide easily. And once your muscle is acting a lot, they add amino acid and build up new filaments, actin and myosin. More filament, big, thicker muscle fibers, and that is called, when you muscle builders build muscle, that's how they build the muscles. Was that the question?
Yeah, because I mean, so yeah, I like to work out and everybody's like, oh, heavy overload, and that increases your muscle size and stuff.
Yep, it increases, but not the number. It's the size of the muscle.
Dr. Shaw, I have a quick question sort of following up on that. In contrast, what happens when your muscles atrophy?
Yes, so when muscle atrophy, again, the same thing, reverse. You will not lose the number of muscles. You still have the same number of muscles, cells. Muscle cells multiply, but in very slow, very minimum rate. As you remember, in the very beginning, we say which kind of epithelium, which kind of tissue can regenerate fast and which cannot. Muscle, skeletal muscle, cardiac muscle does not at all. Neurons does not at all. But skeletal muscle do in some extent, but very few. Once they damage, they multiply and replace them. But when you do exercise, you increase the size of muscle. When you stop exercise, then what happens? You lose all the myofilaments. And you have minimum myofilaments from this diameter, it becomes a smaller diameter, and that's why you lose the muscle mass.
Okay, thank you.
Yep. Okay, now another kind of muscles here, we have cardiac muscle. So we have three kinds of muscles. Three types of muscles are
Cardiac,
skeletal,
skeletal, and smooth muscle.
smooth,
And cardiac.
and cardiac. So some of the differences and some of the similarities among these muscles. Skeletal muscles are attached to
The bones.
bones. That's why we call
The bones.
them skeletal muscle. And when we say muscle and we don't specify anything, it means you are talking about skeletal muscle. Smooth muscle looks smooth under microscope because there is no light and dark pattern. That's why we call them smooth. Cardiac muscle is cardiac muscle, which is found in the wall of atria and ventricles of the heart. And Greek terminology, adjective for heart is cardia. So we call them cardiac. Skeletal muscle is voluntary because it is controlled by somatic motor neuron, which is under your control. Whereas smooth and cardiac muscles are involuntary because they are controlled by autonomic nervous system. Skeletal muscle is called skeletal muscle, and cardiac muscle you see here are called striated muscle because they both have stripes. See here, light, dark, light, dark. The skeletal muscle fibers, as you know, we talked histology a lot. They're long fiber, multinucleated, full of fibers, a single fiber. That's why you call them muscle fiber. Whereas cardiac muscles are branched. You see, this is one muscle cell here. This is branched, and each muscle cell is attached to another muscle cell through a gap. That is called gap junction, and it is called intercalated disc. Yep. That's why these intercalated disc helps cardiac muscle to work in the syncytium. That means if you stimulate one cardiac muscle, then through these channels, sodium, potassium, calcium, other ions can easily spread to another cell. That's why if you stimulate a single muscle fiber of heart wall, that will spread all over the cardiac muscle cell. We're going to talk about that in cardiovascular system in detail. Smooth muscle, on the other hand, they are not a stripe, they are single nucleated, and they are worm shape. You see here, spindle shape. Both ends are tapered and center is ballooned. The pattern of actin and myosin is not like skeletal muscles. These actin and myosins are attached to a dense bodies inside. Okay, they have no sarcomere, they have gap junction and contain 16 times more actin than myosin, allows greater stretching and contracting, and they don't get fatigue easily like skeletal muscle. Actin filaments are anchored to the dense bodies. Smooth muscle contraction is controlled by also calcium, but different from striated because it has little sarcoplasmic reticulum and no troponin, tropomyosin. That's why calcium enters through voltage gated channels in plasma membrane from outside, or extracellular calcium binds with chemodulin. Chemodulin is another protein inside the smooth muscle cell.
Professor, I have a question.
Mhm.
So smooth muscle doesn't get fatigued easily. Is it because it has no sarcomere?
No, they have not the same mechanism. It has not like it is controlled by autonomic nervous system, and it doesn't need all the calcium the same level. It is extracellular calcium. So binds with chemodulin, another protein, and calcium chemodulin complex activate myosin light chain, which is called MLCK, which phosphorylates and activate myosin. Myosin forms cross bridge with actin. This is a complex. I just want you guys to know it. You will not be asked in the exam. Again, that doesn't mean you don't know it. Know it, but you will not be tested on it. Okay? So here you go. How smooth muscle contracts. Cardiac muscle, we're going to talk detail in cardiovascular system. So here, this is the plasma membrane of the skeletal, the smooth muscle. So when there is membrane depolarization, there is graded potential. Once it reaches the threshold, there is action potential. That depolarization in the muscle cell opens the voltage gated calcium channel in the plasma membrane, and then calcium get inside the cell and binds with chemodulin, and that chemodulin then makes the chemodulin calcium complex. There is a myosin light chain kinase enzyme here, which is phosphorylated. Once they are phosphorylated, this is MLCK myosin light chain kinase. When they are phosphorylated, they are inactive enzyme inside the cell. But this calcium complex remove the phosphate, and then MLK becomes active. Okay. So that active MLK then cause the cross bridge activation and contraction. Once there is contraction, then there is dephosphorylation of cross bridge leads to relaxation, and that again dephosphorylation is active process. So myosin phosphatase enzyme, what they do, they dephosphorylate, and again another myosin phosphatase get the phosphate and becomes inactive. So basically there is complex mechanism of excitation contraction for a smooth muscle, and you don't need to worry about this for the exam purpose. Okay, so this is all for today.
Thank you, Professor.
Thank you.
Thank you.
Thank you. Have a good afternoon.
Same to you guys.
Have a good day.
Thank you,
To
Dr.
you
Shaw.
guys. Thank you.
Professor, it said there's gonna be a movie.